Chapter 11 is on blood and blood vessels. And so we're going to be learning the components of the blood. We are going to be learning about the different blood vessels. We'll be learning about stopping blood loss, certain blood diseases, blood types, and then the next chapter we will learn about the heart. So number one, you need to know the functions of the blood. The main word that describes its function is transport, and look at all that it transports. Gases, nutrients, waste, hormones, electrolytes, drugs. It regulates the pH and ion composition. It restricts fluid loss at broken blood vessels because it contains clotting factors. In the blood are white blood cells and antibodies that defend against toxins and pathogens, and blood stabilizes our temperature. Blood facts. It is connective tissue. It is fluid connective tissue. It consists of plasma and formed elements. So what we call formed elements here are what you would think of as cells. We don't call them cells because two of them are not whole cells. There's about 5 liters in your human. Whole blood is the blood that comes out of the body, and then that may be separated into different components. So here is our whole blood, and if we were to centrifuge that blood, all of the blood cells or the formed elements would drop to the bottom. Most of it, 45%, 44% is white blood cells. 1% is the buffy coat. 44% is the red blood cells, and then 1% is the white blood cells, which is the buffy coat, and then the plasma, which is straw colored. And then you can see when you separate it, you can see what's going to come out of the plasma. So this is a good study slide after you've learned all the different components of the blood. So we're going to be talking about plasma proteins and other solutes in the plasma, and then we're going to be talking about the 3 different types of formed elements. Red blood cells are erythrocytes, white blood cells are leukocytes, and platelets are often called, not often because you may never have heard of this word, thrombocytes. So plasma is 55% of the whole blood volume, the liquid portion, and it's 92% water, and it has proteins and other solutes. So the main stuff in the plasma are the plasma proteins. And here are the 3 different types of plasma proteins: albumins, globulins, and fibrinogen. You need to know all 3 types and what they do. We'll start with albumins. Albumin is in an egg. The white of an egg is albumin. So we make this protein. It's the majority of the plasma proteins. And what it does is it allows the correct amount of fluid to remain in the blood. So you've probably seen starving kids. If they're really skinny all over, they're just not getting any calories. But if they're skinny but have a big stomach, they're not getting protein. So their osmotic pressure is off. They're not getting enough proteins in the blood, so water leaves the blood and fills up the abdomen. So albumins allow for osmotic pressure. Globulins have 2 functions. One of them, immunoglobulins, are antibodies, and transport globulins are going to transport lipids in the blood. And then fibrinogen is important in clotting. Fibrinogen is what travels around in the blood, and then when you have a broken blood vessel, it converts to threads of fibrin, and those fibrin threads will trap platelets to form a blood clot. So what were those 3 types of plasma proteins? Fibrinogen, globulin, and what was the osmotic pressure one? Albumin. We have electrolytes. We know these come in from the foods we eat. Sometimes, they're called salts. We have organic nutrients, lipids, carbs, and amino acids, and we have organic wastes. And these are the type of wastes that will be removed in the urinary system by the kidney, organic wastes. The formed elements are about 44-45% of the whole blood. That percentage, 45% of the whole blood, is called a hematocrit. It's mostly red blood cells and a little bit of white blood cells and platelets. The red blood cells are erythrocytes and carry oxygen. The white blood cells are leukocytes and defend against infection. And platelets are cell fragments that allow the blood to clot. So even here you can see that these white blood cells are whole cells. Those large things in the center are the nucleus. These platelets are cell fragments, and whole blood cells are not cells. They've lost the nucleus. What is not a function of blood? Production of red blood cells. That occurs in the red bone marrow. What is not found in the blood? Plasma fats. What is the correct term for a white blood cell? Leukocyte. So our red blood cells are incredibly important. We have so many of them. Here is a picture of a cubic milliliter. And in that cubic millimeter, oh, it's been a long day. In that cubic milliliter, oh my gosh, we have about 5 million red blood cells. A microliter is a cubic millimeter. They talk about red blood cells as biconcave discs. 5 million in that size. And so here's this biconcave disc business. You may recall when you look through the microscope the red blood cells. When we looked at blood, the majority of cells on the slide were red blood cells, and occasionally you saw a larger cell with a purple nucleus, and that was white blood cell. And even here you can see these tiny little dots in here are platelets. This is a cool picture. So this is a sectional view of capillaries, and it shows how as these red blood cells travel through capillaries, which is a capillary is the smallest blood vessel, they stack, and they'll actually stack and squeeze. Red blood cells carry oxygen using a molecule called hemoglobin. Hemo means blood. Hemato, all that means blood. So there are 4 subunits, 2 pairs of twins basically, and each of the subunits holds a single heme molecule, and it's that heme molecule that holds on to an iron ion. It is that iron ion that binds to the oxygen. And when oxygen is bound to the iron, hemoglobin is called oxyhemoglobin. It is bright red. When there is not a lot of oxygen, it's a dark red, and it's called deoxyhemoglobin. You know, in the textbooks, they have red vessels and blue vessels, and the red represent arteries. We do not have blue blood in the veins. It's darker red. So when you have blood withdrawn, that's that darker red. We're taking blood out of veins, not arteries. So red blood cells are produced all the time, 1% replaced every day, 3 million new red blood cells every second, but they don't last for long. They last for 4 months, and then they're going to be recycled. They'll be recycled because that iron in that red blood cell is incredibly crucial to the body. So either that red blood cell will be phagocytized by a macrophage, or it will just erupt and it will be broken down in the liver, the spleen, or red bone marrow. What is the average red blood cell count per millimeter cubed, which, by the way, is also a microliter? 5 million. What pigment carries oxygen in blood? It is hemoglobin. What is hemoglobin called when it is carrying oxygen? Oxyhemoglobin. So now we want to talk about blood types. You may or may not know your blood type. On our red blood cell is a protein, and that protein is called an antigen. And the antigen triggers an immune response. We have these antigens because it's one way of our cells recognizing cells that belong in the body. So if you put a blood cell with the wrong antigen in our body, we recognize it as foreign and we attack it. So what's going to attack it will be antibodies. So your blood type is determined by these antigens, just a little protein that hangs out on the surface of the cell. And the most important are A and B and D. Now, you know there's A blood, B blood, AB blood, and O blood. O blood, those people have no antigen. D is the Rh factor, and we'll talk about that in a second. But if you have A blood, your plasma contains antibodies against B. These just develop naturally. So here is our A blood, and here is our antigen for A. Now naturally you're going to have antibodies that are against B. So if you have B blood enter into your body, these antibodies will fight it immediately and clot. Type B blood has surface antigen B, and the person has antibodies against A. If you have AB blood, you have both A and B surface antigens, two different kinds of proteins, and you have no antibodies. And if you have no antigens, type O blood, you have both antibodies. So should you be exposed or get a transfusion of blood that is the wrong type, those antibodies will cause a clumping of the blood cells, and this clumping is called agglutination. And then those cells will die and you'll die. So you don't want that to happen. You've heard of positive and negative, Rh positive, Rh negative. That's just a completely different antigen. Sometimes it's called the D antigen. Sometimes it's called the Rh antigen. So if you have that surface protein, you're Rh positive. If you don't have it, you're Rh negative. So your full blood type is reported as a letter or two letters and a positive or negative sign. So if you look at the distributions around the world, blood types are a little bit different. In the US, the majority of people have type O blood, average 46 for type O, 40 for type A, 10 for B, and 4 for AB. So AB is the rarest. In Australian Aborigines, they have no AB or B blood. So if you were in the lab, we would be doing using these cards called Elgin cards, which can test your blood type. So on the card is a little bit of the antibody, and you put a drop of your blood there and mix it. And if you see clotting, you know that um, is, you can determine what type of blood that you have. Um, if you were to receive the wrong type of blood, it's called a transfusion reaction. And so therefore, donors and patients' blood are typed to find out exactly what blood type they are, so there is no transfusion reaction. This is about what an Eldon card would look like. You see this clotting going on here. Type A blood. Okay, so this is anti-A says there's antibodies against A here. So you put A blood there, and if it's A blood, it will clump. With B blood, there's no clumping because this is A blood and it's anti-B antibodies. Now here, there's clumping with the Rh, so we know it's positive. So anytime you see clumping, it's positive for that blood type. Here's B positive blood, here's AB positive, and O negative. As I said before, the antibodies develop. I've read in some books that it's present at birth. I've read in other books that it develops after birth. And then finally, we'll talk about the hemolytic disease of the newborn. This deals with the Rh antigen. Now, the antigens are genetically determined. A child will inherit genes from both parents and may have different blood type than either parent. During pregnancy, the mother's antibodies may cross the placenta. So if the husband is Rh positive, or the dad is Rh positive, and the mother's Rh negative, and the child just happens by chance, 50% chance of being positive, if the fetus is positive, the mother's anti-Rh antibodies can attack the fetus. And this is called hemolytic disease of the newborn. So this Rh factor is important for women who are Rh negative. Now, it's not important for the first baby, but for the second baby and third baby and so on. This Rh mother Rh negative mother will not develop antibodies until she's exposed to Rh positive blood. So during delivery, some of those fetal red blood cells may mix with her blood cells and causing her to produce anti-Rh antibodies. So future babies may be in danger. And so we give a shot called Rogam. You may have heard of it. You will have it prior to delivery. You'll have it after delivery. This erythroblastosis fetalis, when the fetus is Rh positive and the mother's Rh negative, is fairly fatal without treatment. So this Rogam protects the fetus. And so the only problem you're going to have is if the mother is negative and the fetus is positive. During childbirth, some of these cells can pass into the bloodstream and she will develop antibodies against Rh positive. If the future baby is Rh positive, her antibodies will attack the fetal red blood cells. What is true about type B blood? Two of the above are correct. It contains B antigens and it contains anti-A antibodies. When is the Rh antigen a problem? When mom is Rh negative and fetus is Rh positive.